All right, guys. Thanks very much. This is my third time at camp. I'd just like to say that it's always a privilege to come here and to talk to you about how science might be able to advance some of the clinical care in DC. So I'm going to speak slowly. It's going to be a challenge for me as well. Very careful, but I'll try to remain calm. The main things I want to go over with you today are, I was asked to talk about telomere biology, and one of the things I realized is that you already know so much of it from the past couple of days. And then the other aspect is going to be to talk to you about new research, technology. So you've heard about what we can do about the blood. You're hearing from hematologists. There are a lot of things that we cannot answer in terms of what happens next, and that's where a lot of the studies that you're doing via SEMA and NIH and other folks are going to do to try to answer what happens when you actually look at all the patients with DC. But the other part that we need is, what do we actually do about all these organ failures? What do we do about these tissue failures that we have? And I want to give you a glimpse at some of the technology that's being developed in biology in general, and how I think it actually is really applicable to DC as a prototypic disease as well. Sorry about the technical difficulties. So we're going to go through telomere biology a little bit, but it's going to be really a review of telomere biology. And since you already heard it, it's going to be very interactive. I'm going to call on you. You're not allowed to look at your sheets. We're going to find out what you've actually learned in the past couple of days. And the next area will be just talking about new research. A bit of an update from the last meeting of what is advanced. We'll talk about genetics, models, and technology, and a little bit about how, even besides what's going on in the lab, how we're trying to get organized and answer questions together, both on the research side and also on the clinical side. So, consortium update. All right, so the cell, the basic unit of your body. And we're interested in the middle part here. What's going on in the middle? So, what's that thing that the text called? The nucleus is what houses it. What's that? Chromosome. I heard chromosome. That's right. Okay, very good. So we're interested in the chromosomes and a part of the chromosomes in particular. What are chromosomes made up of? Genes. DNA, genes, exactly. And so DNA is basically 3 billion base pairs of DNA that are in 46 chromosomes that code for all the proteins, as Dr. Savage and others mentioned to you. And these are basically the building blocks of how all your cells work. And then beyond that, are we more interested in the chromosomes and the DNA? Telomeres, right, exactly. That protect the ends of their chromosomes. So on each chromosome, this is one chromosome out of 46, and the ends have these telomere repeats that are found here. And that's very important in this area. And we think about it in some ways as the end of the shoelace that protects it from fraying as it goes down. Anyone know what the bonus point? Anyone know what the name of that is? The end of the shoelace? Excellent. Okay. A veteran. Randy. Okay. So this is the protective structure of a long string, and the same thing can be thought of as a telomere as well. So telomeres, they shorten when cells divide, right? Every time the cell divides. So this is basically a telomere. This is a schematic showing if you have a cell, every time it divides, the telomere is getting shorter and shorter. And at a certain point, the telomere will reach to a point where the cell wants to stop dividing. And that's when the cells can't proliferate anymore. And you need cell proliferation to maintain your body's tissues throughout life. Different cell types, different tissues need more division compared to others. So what is the thing that actually lengthens telomeres to keep cells dividing? Telomerase. So this is a machine that's pretty amazing. It's basically a protein, the telomerase reverse transcriptase, and RNA component. And this basically serves as a template, meaning like the code that adds more and more and more repeats as time goes by. And if you have that, then when you have telomerase, even though the cell is dividing, the telomere end will be maintained and it keep dividing and dividing as well. And so this was an amazing finding in biology. This is true about all cells, and this was awarded the Nobel Prize to Ryder, Blackburn, and Drozdak in 2009. So you'll see also that these are actually very fundamental biological principles with really wide-sweeping implications that actually form the foundation of this disorder, this DC. So just to recap, then, this is human telomeres. The other one was tetrahymid. I've noticed everything was figured out in like little one-cell organisms, but this human telomeres are composed of these repeats, TTAGG. They protect chromosome ends. The length correlates with the number of times the cell can divide in the absence of telomerase. telomerase in some cell types, stem cells, embryonic cells, cancer cells, and keep the cell dividing by extending the telomeres. And these are the subunits of telomerase. Here's the RNA component. Here's TERT, the reverse transcriptase. Here's these accessory factors, several of which are mutated in Dyskeratosis congenita. These components are actually in all cells, but they can't function to elongate telomeres without this component, which is actually present only in the stem cells. And I'd like you to remember, just for no reason at the moment, these letters right here. So this is the template for telomerase, the RNA template CCTA, TAA, CY. The other thing that protects the telomere end is not just the length of the repeats, but also the structure that you've also been introduced to. This is the T loop, so the telomere end goes and wraps back upon itself. And there are several proteins that protect it called sheltering. Some of these components are mutated in DC, TIN2, TPP1, which Dr. Savage and other groups have shown. So telomere ends have a complex structure, which is important for their integrity and structure. And I'd like to remember this structure here. So once again, these components are all mutated in Dyskeratosis congenita. So these components that are actually part of the sheltering complex, these components that are part of telomerase, and then other components which are important in replicating this, capping this end, and also taking this whole structure to the proper place in the cell where telomeres can be formed. So these are the colored circles that indicate the genes mutated in Dyskeratosis congenita, again, just as a review. And so you already know patients with DC have shorter telomere length in their cells. So this is a patient with DC. This is a blot. So basically you take the DNA and you spread it on something, and the shorter ones run farther along. And you can basically probe for it. So this blob is like the distribution of telomere lengths in the cells, in the blood cells of patients with DC. These are the parents. So this is how we study it in the lab. These are the parents. They have longer telomere length. So this is abnormal for this person. The other way of measuring it is very pretty, really cool way. And you take a cell that's about to divide, it has all the X's that you're familiar with, and you can actually hybridize these glowing probes of a different color to the telomere end, and you can actually quantify how long the telomere is by how bright the signal is, and assay all of those numbers together to see what the distribution of telomere length is. And this is the basis in a slightly different way of this test, which is actually very, I think, has been extraordinarily important in this disease. This is a Blowfish Test, which basically takes a sum of the fluorescent signal, like glowing signal, from telomere ends in the cell, and basically plots it according to what normal would be. So this is normal. So this person would be at the average length of telomeres in this particular type of cell compared to all the people who are the same age in the population. This person would have a telomere length that's greater than 99% of the population. This person would have a telomere length that's about the lowest first percentile of the patient, and this is a DC patient, which is way below the first percentile line. So this ends up being a useful, very useful diagnostic test for DC. So how does this impact how cells divide and form? You have already reviewed this to some degree. Several cell types in the body require constant replenishment on a daily basis to actually maintain the function of the organ. Some don't as much. They basically have cells that are there and they function for days and years without actually undergoing damage proliferation. The blood is a great example of a cell type and a tissue type that actually undergoes massive amounts of proliferation. This is a blood stem cell, and from what all we know, there's actually a type of stem cell here that in itself doesn't have any function. It can't do the things that it needs to do. It actually can divide and replicate itself over and over and over again because it has some telomerase in it, and it can basically last for life. And when it gets the right signals, it basically can differentiate into various cell types. So the ones that you're familiar with are the track, are the platelets over here, the red cells over here, the neutrophils, lymphocytes. So these are being produced, like Dr. Petrucci said, at the level of billions to trillions per day. That's pretty amazing. And you can actually have this stem cell divided to just a few numbers to give rise to these billions because of amplification of these cells at every one of these stages as well. So you can imagine that in this cell type, this is a normal bone marrow, composed of many, many of the stem cells and all the cells along that lineage, giving rise to the cells that are going to come out into your blood and do their function. This blood cell, this whole factory is dependent on the ability of the stem cells to self-renew. And what's happening normally is that, sure, there's stem cell telomere shortening, but there's lengthening too, so there's some balance going on. But in the presence of a telomere gene lesion, we think that in the blood system, there's telomere shortening over time with division, and at a certain point, instead of being cell types that can last forever, there's basically loss of this ability to self-renew to a sufficient stage, and also then you have the onset of bone marrow. And so this possibly is happening in other tissues as well. We really don't know. We don't actually have a solid idea of whether this is the only way that the bone marrow is failed in DC, and also then how it's actually happening in other tissues. What's the reason for all of these vessel problems in the eye, and the lung, et cetera, et cetera. And we really need better ways to actually study it. So the goals for research in trying to make some advances here have been all along diagnosis, which has gotten so much better in the past 10 years, disease mechanisms, which we still actually don't understand that well, and then the translation of these insights into new treatments. What new things can we get out of the lab that are based on the new biology that we learned about DC? And that actually haven't heard too much about brand new drugs that are specifically for lengthening telomeres or anything yet. It's just that we just don't have that yet. So the different ways, in general, you try to study a patient's disease. You need tools. So the researcher needs things in their hands to actually work with. So one of the things that's very important is to understand the genetics. So that's actually been very successful in this work. If you know the genetics, then you can do things like make animal models. I'll describe this in more detail. It might seem a little bit odd to you, but this is fundamentally important in the research lab to actually try to create the same sort of disorder in another model so that you can actually understand how it affects the whole body. You can't just, not just looking at the cells in the lab. The other is to actually study cells from patients. So a lot of you have actually donated cells to the lab, and I can really tell you that's an incredibly valuable thing to do, because even at the end of the day, you'll of course appreciate that animals are not humans, and the particular genetic mutations that patients have in DC are very, very hard to model. So we get blood cells from patients, skin cells from patients, and try to study them. I'll tell you about another technology to try to engineer these cells because of some limitations into other cell types, stem cell types. You take all of these different tools that you have and try to understand disease mechanisms in the lab. Try to think of treatment strategies based on these disease mechanisms, things that new ideas that actually could be used for intervention. Test your treatment strategies first in your models that you have in the lab, cell types, engineered cells, animal models, and then if successful, this is the basis of actually forming a new therapy for the patient. So, new research in DC in the past 2 years, so there have been 2 genes found. There were 8 in 2012, now there are 2 more. Does anyone remember which ones they are? ACB, really very good. Okay, so RTL1 and ACB, which codes the protein TPP1, so 10 genes. This is now accounting for almost 60 to 75% of the cases of DC, depending on the cohort. And that's actually, again, remarkably important. Still all converging on telomere biology, and this has been very, very helpful in order to give hints of what things to actually modify inside animals and cells. The other thing that's actually, I think, a big innovation that was already touched on in the category of diagnostics and genetics is the idea of getting your whole genome sequence, even to discover new genes, actually nowadays as a more streamlined way of sifting through those other 10 genes too. It's actually hard and cumbersome to go gene by gene by gene. So this is something that you should appreciate, that in 2001 it cost about $100 million to sequence a genome. And what's actually happened over the course of the years, very precipitously with new technology that's become available, is that this has come down exponentially to the point where sequencing a whole genome is now somewhere in the $4,000 or $5,000 range cost. If you actually sift out just the portion of the genome, the 1 or 2% of the genome that actually codes for any genes, that cost on a research basis can be less than $1,000. So that's truly revolutionary. And what's actually happened is that this type of analysis of the coding portion, in some cases of the whole genome, has actually now become a clinically certified test, which was not true even the last time, as widespread the last time that we spoke. So this is something that you should just know about, that sequencing the whole genome is becoming very tractable. And now the limitations aren't even in the technology of sequencing, it's more in the analysis and the amount of information that's gathered. So how about disease models? So we and others, of course, have been able to get samples with the guidance and research participation of patients. Ideally, the problem is in blood cells, like you study the blood stem cells. If the problem is in other cell types, like you study those, but of course that's really hard to get. It's hard to get lung stem cell, et cetera, et cetera. It's hard to even identify. The other problem in DC is that whereas other patients' cells can actually grow for some period of time in the lab, like we already talked about, cells from patients with DC actually tend to clear out pretty quickly. So you don't actually have that much material to do work with. And in general, when patients come in, sometimes they already have bone marrow failure, so it's really hard to even analyze their blood system. This is exemplified here. So when we collect fiberglass samples, so this would be a skin sample. So this is basically something that's growing out of the size of a biopsy that's the tip of a pencil, like the lead that you can see in a sharpened pencil. So it's a relatively small biopsy. And so this is that part of the skin underneath the microscope. And out of it grow these cells, each one of these blobs is a cell. And we can grow them and grow them. And from someone who doesn't have DC, they grow quite a bit. And you can get millions and millions of cells from an unaffected person. But the DC cells actually really do have a hard time growing. They basically stop growing after a very early period of time. So you only can get a certain number of them. And so I do this still myself. I can't even really trust my lab people to do this, especially when I get them from a patient with DC. I really actually want to expand them as much as I can and protect them and archive them as well. And over again, I'm faced with this issue where a regular cell type will grow and be happy and have this nice morphology in the DC patient cells. When they're starting to get up to about here, start showing that they actually don't want to live anymore. They kind of want to die in the culture and stuff. So the key here is to actually try to immortalize these or turn them into something else pretty quick. So once you know the genes, and once you have some cells, even some cells, you can basically, there's one type of concept that allows you to actually go a little bit further, and that's about using pluripotent stem cells. So I can define what that is for you. That helps you make animal models, and that helps you make stem cell models for the lab. So pluripotent stem cell model. So going way back to embryology. So this is a mouse 2-cell embryo. So the mouse sperm and oocyte have fused, they've turned into one cell, and they start dividing. 2 cells, 4 cells, 8 cells, etc. And for a while, they're just these collections of cells that don't really have any definition. And soon they start to specialize. They turn into this thing called blastocyst. Okay? And the outer cells are going to become the placenta, and the inner cells are going to become the mouse. So what was discovered about 30 years ago is that you can actually take this cell mass and grow it under very, very specific culture conditions. So this was quite revolutionary, where these cells are in a suspended state of animation. They basically think that they're still in their cell mass. And they'll just grow and grow and grow. And one of the things that's the most important about that is they think that they can make anything in the entire body. They're basically responsible for making the entire mouse. So you can grow them and grow them and grow them. So these are called mouse embryonic stem cells. If you introduce these then at some later time into another one of these blastocysts, they can go on, these cells can go on and give rise to any tissue in the body. So that's what pluripotency is. Basically being able to isolate a cell that has the ability to become anything. In addition to this technology, there was a technology that was relatively inefficient but feasible to actually change the sequence of any gene that you wanted anywhere in the mouse genome. So all of a sudden it became possible to actually generate a mouse carrying any genetic mutation. So that's why knowing the gene, and knowing the gene in the human, and knowing that mice have almost all the same genes that humans have, you can basically then isolate and target that gene for modification and create a mouse, an animal model. And so this was awarded relatively late, actually. This study was done in the early 80s and 90s, and this won the Nobel Prize in 2007. It's been incredibly important to understanding human genetics and disease. So it's been very hard, actually, to model Dyskeratosis congenita gene lesions in mice, even using this method. And it's because, as Dr. Savage showed you, the types of mutations that occur in patients' genes are this modification here, or that modification there, in a rather complex way. They're not necessarily usually just obliteration of the gene itself. And we think that that in the human and in the mouse frequently actually just doesn't allow for the production of a real mouse. So it's much harder to actually engineer the precise mutations that patients have in the mouse. And then, of course, there's the matter of who's going to undertake that, who's motivated enough to really do such a thing and make a proper model. But that's been happening because there's enough. I'd say that one of the things that has happened in the past 2 years is that there's been a few models that have been made to actually try to mimic truly Dyskeratosis congenita lesions. So one of them, this gene has not been shown to be mutated in DC yet, but it's part of the sheltering complex. And a conditional knockout, meaning they were able to knock it out just in the blood system, was able to show that the mice got bone marrow failure. One reason why mice don't actually mimic telomere biology, not in addition to the fact that they actually have these alleles, these gene types are actually hard to make in the mouse, is that they actually happen to have really, really long telomeres. So if you actually get rid of telomerase overall, you can basically breed them for 5 generations and they actually still don't have any problems. So there's differences too. Mice are not humans and vice versa. So this has been a little bit of a difficulty, but people are trying to approach it using mutations of other genes and actually really interested in seeing what happens. And this is just the beginning of that. The other thing I have fish up here is that actually there's been a lot of development of the fish as a model. And this is a zebrafish, and it actually happens to have telomeres that are much more around the same length as humans. So there have been a few studies now trying to take telomere gene mutations and put them in the zebrafish. And this is one of the papers that was published just last year, of seeing some phenotypes, meaning some disease manifestations in a fish model of telomerase mutation. Now, I know that this sounds very odd, I'm sure, in a lot of ways to you, but this is really the only way of actually getting at how these things at a physiological level actually cause disease. The other one that I think is actually impressive is this, because this is actually an effort to really model a human mutation. So this is TNF2, a TIN2 Dyskeratosis Congenita mutation, which was trying to literally model one of the human alleles, the K280 mutation of TNF2 in a mouse. And basically what these investigators found was a more impressive phenotype than trying to nullify the telomerase gene mutations. It didn't take 5 generations to actually see some phenotype. So there's some inroads being made in this, and I know that there is effort to create other models specifically of DC genes, but it has not been easy. The next technology to tell you about where we're shifting from animals to actually cells, to using human cells, is how do you make a stem cell model? Like I was telling you about a pluripotent stem cell model, one that can make any tissue of any kind from a regular sample that you get from a patient. So remember, going from a one-cell embryo into the whole body, the cells have specialized all the way along. The eye cells are eye cells, the skin cells are skin cells. And it was thought for years that that was a point of no return. You couldn't actually go back to being like an embryonic stem cell. But there's been a tremendous amount of research in the past 10 years in trying to understand that if the original cell has the same DNA as the skin cell, even years and years later, why can't it be the same? What are the things that are different about it that actually don't allow it to be the same? And this individual, his lab, did a remarkable experiment. They were studying what made these cells look like themselves, what genes they were expressing. And he took some of those genes and put them back inside the skin cell of the mouse. And then by doing a few manipulations, he was able, over the course of 4 weeks, to actually turn this skin cell back into one of those embryonic cells that actually had the potential of turning into anything in the body. You can realize the implication of that. And to prove that, basically, these cells that used to be skin cells were put back into an embryo and turned into a mouse, and turned into a whole mouse, essentially. So it truly had retained all of the capacity to form an entire animal if it was just put under the right conditions in the lab. Okay? And so for this, very quickly, he won the Nobel Prize 6 years later, and rightly so. This was a remarkable demonstration. So very quickly after this demonstration in the mouse, there was tremendous interest in seeing whether this is the type of thing that could actually happen in the human. And sure enough, basically by using the exact same approach, you can take skin cells from humans, introduce these genetic factors, all sorts of stuff happens to the cells. They turn into different things. Some of them grow, some of them don't. But you can actually see the ones that look like human pluripotent stem cells and isolate them and grow them and grow them and grow them. And now they have several things. They've turned on telomerase, so they have a trait. They will grow forever, and they can actually be turned into many, many different cell types inside the lab. Okay? And you can't do the test, of course, of whether they can make a whole human. That's completely not allowed. But to whatever extent you can, you characterize that these cells have the potential to form any form of tissue. So the utility of these cells became evident right away, is that if you take a small sample of cells from a patient, you subject them to this process, and isolate these pluripotent stem cells that are carrying the mutations of patients. Now, you don't have to worry about differences from patient to patient. You've got specialized stem cells for each patient. Now you can study what the effects of that mutation are on stem cells. You can also try to turn these cells into other cell types and see along the way how that mutation is affecting the formation of nerves, blood, and muscle. And then, of course, if you can actually take these cells and fix them in some way, turn them into these tissues, then the holy grail is whether you can actually turn these into some form of therapy. You can expand them into food, so, which is something that you guys were talking about a little bit yesterday. So here's just a demonstration. These are skin cells from a patient. You turn them into iPS cells. You subject them to other conditions and differentiation conditions and factors in the lab for 2 weeks. You take these things that form called embryoid bodies, you subject them to other factors, and all of a sudden they're starting to turn into blood. So this is still on the microscopic scale. You're basically seeing it inside the dish. But they're starting to form from skin through this intermediary, basically these cells that look like and function like blood cells. But these still, of course, carry the mutation of the patient. So the other thing you need to be able to do then, if you're going to turn it into a therapy, or if you want to compare what happens when you have a normal versus abnormal for a single patient, is manipulating the DNA of the cells, right? You want to actually fix it or change it in whichever way you like. So this has still been very hard. I showed you how it was being done in the mouse, but it's very inefficient. And so there have been tools, and this is truly a technology that's developed in the past 2 years, to actually more easily manipulate the genome of any cell type, in any animal model or any human cell type too. I'll mention these, you don't have to memorize any of this. These are technologies called Talons and CRISPR-Cas9. Most labs can adopt this. It's not very hard to do. You basically get this off the shelf, learn quickly how to do it, and basically apply these technologies. These come from bacteria. It's pretty amazing. These technologies are coming from understanding bacterial immune systems, essentially. And there's a protein. It uses a piece of nucleic acid, like the telomerase RNA component is, an RNA. And this RNA guides it to any place in the 3 billion base pairs of the genome and tells this enzyme to make a cut there. And so why would you want to make a cut there? So you can make a cut there and actually disrupt the gene. So you can create a particular null or a disrupted gene. But even more interesting is that what the cell knows to do is that when it actually gets cut, if you put in another piece of DNA that you want it to swap out, that process becomes much more efficient. So you can actually trick the cell into swapping out the DNA that you want for the DNA that it has inside its genome. It's much, much more efficient this way. It's still technically challenging, but it's actually very approachable at this stage with cell lines, etc. So this is something that actually has become, you might hear about this, part of the reason I'm mentioning it, this ability to engineer the genome in human and other cell types. So we did this, just to give you an example, using a telomerase RNA gene because we're interested in DC. So here you have the same thing I showed you before, similarly a plot of a human cell line. And it basically divides and divides. It has telomerase, so its telomeres don't really shrink. They just kind of stay along the way, and it has normal copies of TRK. And we put in these genome engineering tools and killed the copy of TRK, both copies of TRK that were in there. So it no longer has the telomerase RNA component. So you can see that this cell basically starts to lose its telomere length and lose its telomere length, and finally you can't see them anymore, and the cells stop growing. They die in the dish. And then to prove that we could actually do it, we basically put in the same modifier, the talon in this case, and also a new repair construct to basically fix it and restore the copies of TRK. And then the cell, if it's over here and it's on its way to dying, which we actually fix it in that interval where it's still dividing, we actually can rescue it and it basically grows up. And even in this case, of course, when it's only happening in 1 in 100 cells, for instance, all the other cells are dying. So these are the only ones that actually grow up. So this is very achievable, and we want to use these types of approaches for different mutations and cell types and engineering these in human cells, and especially in applying this technology to the iPS cells. So the goal here would be now to combine these technologies, take patient cells, skin cells, turn them into iPS cells, try to fix the mutations, or alter the genome in other ways, and see how that works. You can study these cells themselves, but also then ultimately in a therapeutic scheme, which is still not reality, it's basically off in the distance, is try to actually turn them into tissue-specific stem cells, so ones for blood or other tissue types, and then basically put them back in the patient. So the biggest barriers at this point are, anytime you're doing cellular manipulations, is safety and genetic integrity. Is the DNA all sort of the same? There are protocols being developed for turning these cells into this cell type and that cell type, and ultimately, for blood, all you need are cells and you put them inside the patient. But if you need an esophagus or a lung or something like that, there's a whole other aspect of this about tissue engineering and creating a complex organ, etc. So you can see that there's a huge amount of complexity to this, but some of the tools are available to try to approach these problems. Okay, so now I'm shifting gears. So that's the future, what we can do from the lab. But even as we basically think about the future and what can come out of the lab and ongoing efforts using patient data, patient materials, etc., technologies in biology in general, there's still an impetus right now to try to do what we can with the information that we have and advance research that impacts the care of patients not in 10 or 15 years that you'll always hear about this technology, but more acutely. So we have initiated a research consortium, which you'll recognize this now. So you know what it is, right? That's a Telomere subgroup, right? And this is basically the T loop there. That's why I do pay attention to this. But anyway, we made this acronym, the Critical Care Consortium for Telomere-associated ailments, really to keep it very broad, but the purpose of this group was actually to address a variety of telomere diseases. You've seen that there's such a spectrum. And we've been meeting, we met twice, most recently in Chicago at the end of last month. And the goals really, so first I should define what a clinical consortium is. So this is a group that's, so consortium in general is a group formed to undertake an enterprise beyond the resources of any one member. Okay? It can be in any context. But in a clinical research context, one of the reasons that we are trying to create this is to overcome barriers, academic barriers, clinical barriers, whether they be because of individuals wanting to do things their way, perhaps, or just because there isn't enough communication. We're trying to improve communication and actually improve the opportunity for collaborative research, especially in a rare disease. And the overall goal of this group is not so much to do basic science research together, but to improve the care of patients with DC and related telomere diseases through collaborative clinical research efforts. So the goals of these things go very slowly, but I think we had identified some goals of the first meeting we had in Chicago, was to end up, first and foremost, with some sort of multi-institutional treatment trials for patients with DC and related disorders. And I should just pause for a moment and explain why I think when you're doing trials, what does it mean? Because you go to different centers and you actually engage in different therapies, so you have a certain disorder. On the one hand, your doctor might say, well, I think we should just try this medication for this disorder. That's, I'd say, at one level. At the other extreme is that there's a trial going on that's international, that's basically involving many institutions, where many people have thought very carefully about what interventions should be made for that particular ailment, how we're going to compare and analyze the data, so that ultimately there's some real information that comes out for patients. So the difference between doing it for one patient and not really reporting the data necessarily versus actually coming up with some real information that advances the field and advances the care of patients in general. That part is very, very hard to do. And so the goal of this group is to actually facilitate that. The other goals of this meeting were to strengthen international collaborative efforts that are definitely growing, I think. So a matter of actually getting researchers from around the world to actually all think about the disease together. And to facilitate, this was a side benefit, was to basically say, if we're getting all these people into the room together, you've been hearing about the DCO guidelines book for the care and management, that if all the folks are there anyways to facilitate them, actually as a separate effort and initiative, reviewing all of these chapters together and discussing them to sort of make them come up with some consensus. I'll tell you who joined us. So it was relatively successful. Overall, we had 28 participants from 19 centers in 6 countries. I'd like to highlight that several of the people here, and also from DCO, were actually present at that meeting in Chicago, and they went specifically just for this meeting, which is important to say. And especially when we consider the folks who were flying in from international locations. We had Agarwal from Sydney, who some of you know. We had folks from Germany, Toronto, Italy, and Jerusalem who joined us. There was a fire at the FAA tower in Chicago the day that this was actually happening. And so some people actually couldn't make it. In fact, we thought that the whole thing was going to fall apart at the last minute. And so a few people couldn't make it, but it was quite remarkable that this many people actually did make it. Considering that the flights were shut down for like the entire tri-state area. I had to drive from Cincinnati, for instance, which was interesting to drive to Indiana. But overall, we had a very good discussion, and we identified some major initiatives, just to let you know. So again, this is relatively early. One of the things we wanted to lead with some goals for the next year or so. One of them was to improve the diagnosis and definitions of DC and related disorders. As you see, the genetics is telling us that there's a huge spectrum. Even the doctors can't agree on what to call what, right? And that's important for the patients, that's important for the physicians, that's important for designing studies and determining who's in the category and who's not in the category as well. So this is an effort to actually refine the diagnosis, determine some definitions, and also to serve as a place, as a group of experts where people can call in and say, what do you think of, ultimately, what do you think of this set of data, this genetic finding, et cetera, to try to refine the diagnosis. That's what some folks in this group will try to do. Second, we identified something very specific that we wanted to pursue early on. And this is basically, amongst the members of the group, pretty early on, it was becoming known that there were patients that we were taking care of who had pulmonary arterial venous malformations that Dr. Bertucci talked about. And amongst the email trail of this group, it became clear that there were actually many more people who were suffering from this than we thought. And we thought it was a good platform to try to make a little task force, essentially, of this group to gather the experience of these patients and describe the problem better for the literature. And also to try to come up in real time with something that would be an interventional trial. So can we understand the biology and know exactly what to do about this and come up with a specific intervention in the next year or so? That would be very, very hard. It might take years to figure out what the biology is. But there are patients right now who actually are looking for an intervention, and some physicians who are going to do that thing where they treat off the shelf, which is nothing wrong when there's nothing else to offer and the patient really needs something. But the ideal way of doing it would be to say, a group of people have thought about it, this is a reasonable type of intervention, here's how we're going to study it, here's how we're going to study the outcome. So I think that that could be a goal of this group as well. And then a major initiative near and dear to my heart would be to actually come up with a bone marrow transplantation, one or more multi-institutional trials. So there's the status here. You've heard in a very nice talk about Dr. Blood about all the different considerations around bone marrow transplantation and how it went from being just doing it the same way we did it for everyone with aplastic anemia to trying to tailor it more and more and more for patients with DC and hoping that the outcomes would actually improve. And there's been a lot of progress in that area, especially from the first prospective trial that was a single-center trial from the University of Minnesota started by Dr. Jacob Talar, who's on the medical advisory board here. And that's actually been very, very important. It was doing something where you say, this is how we're going to do it for all of our patients coming through. We thought about it, how we're going to do it for these patients. Let's see what happens. Is definitely advancing the field over basically just doing it this way or that way, kind of, not randomly, but without actually collecting data appropriately. So that has actually fueled a lot of interest in this area. We now have an open perspective trial as well. And the time is right, I think, for groups to form around a particular idea. It may not just be one, but a couple of ways of actually approaching that are sensible to DC trials. And there's a lot of appetite for this. There's a lot of interest in this sort of thing to do a multi-institutional perspective trial. So these discussions are ongoing about how to move forward. And this will be very impressive. This will move faster than it has, even though FA was organized a long time ago for disease like anemia. And part of it is overcoming, like I said before, academic barriers. Like we do things our way, and that's just the way we do them, and basically having people work together. I think that there's a good spirit for that. The other thing that's very important is this question of what happens after. What happens after your bone marrow transplant? What should we do next? How are things going to progress? And that's not being collected in a very, very careful manner. Even if places do transplants in a different way, there's actually an opportunity perhaps for seeing what the long-term effects and the natural progression of the disease are compared to what happened after the transplant. And that's actually very important for prognosticating. A lot of people have questions after their transplant. What is going to happen next? What was the effect of this agent? What was the effect of the graft versus host disease, et cetera, et cetera. So what we're hoping to do is to look back and try to collect the data on patients who already had transplant, but even better than that. So that'll serve the needs for now, but even better than that to look forward and see what's coming afterwards in the context of these trials. So just in summary to wrap up. DC, as you know, is one of a type of very rare telomere diseases. It's been a challenge to diagnose, study, and treat, but I hope I can make to you that there are major strides in the genetics and functional tests and general awareness. We have a lot of work to do in the lab and otherwise to understand how my parts of the body are affected. And we have new tools and structures. So these are organizational structures to overcome limitations of patient numbers, materials, animal models. And I think there are really disease-specific treatment trials coming for this disease, which is unique compared to other disorders as well. I just wanted to echo what Barbara was saying about spreading the word. I think that this organization deserves a tremendous amount of credit, formed in 2006, and having had just 3 camps in spreading the word about this disease, garnering interest, and actually helping patients literally around the world. The physicians are starting to get organized too, so Sharon and Dr. Riddell organized a conference in Sydney just this past June. We've organized this as well. Part of these interactions led to the webinar that Barbara mentioned, and you have your initiative that's growing, which will definitely interact with these groups groups as well in terms of facilitating research in the future. So thank you.